This presentation is about the role of GABA and chloride ions on the body. Firstly, let's look at what GABA is. The full name of GABA is GABA aminobutyric acid. GABA is the main inhibitory neurotransmitter within the central nervous system. The primary role of GABA is reducing neuronal excitability throughout the nervous system. I like the analogy in the picture saying that GABA is like the brakes in a car to slow down the brain activity and help us relax and maintain a sense of calmness. So where does GABA come from? GABA is secreted in various sites of the central nervous system, including the cerebral cortex, hypothalamus, the cerebellum, spinal cord, granule cells, and the retina. GABA is synthesized from another amino-acidic neurotransmitter called glutamate in a reaction catalyzed by the enzyme glutamic acid decarboxylase. GABA is considered to be a multifunctional molecule where neurons in every region of the brain use GABA to fine-tune neurotransmission. There are two types of GABA receptors, the inotropic GABAA receptors and the metatropic GABAB receptors. When GABA interacts with the receptors of a neuron, this inhibits the generation of action potentials, therefore impeding the ability to excite nearby neurons. When GABA binds to the GABAA receptor, as shown in the picture I have drawn on the top right, this results in the opening of an associated ion channel, which is permeable to the negative charged ion chloride. When these negatively charged ions flow into the neuron, they hyperpolarize the membrane potential of the neuron and decrease the likelihood of neuron firing an action potential. When the GABAB receptors are activated, as shown in the bottom right picture, they frequently cause the opening of potassium channels. These channels allow positively charged potassium ions to flow out of the neuron, also making the neuron hyperpolarized. Again, this decreases the likelihood of the neuron firing an action potential. Increased GABA activity can have sedative effects due to the reduction of neural transmission. Certain medications can enhance the effect of GABA on GABAA receptors. Benzodiazepines, such as Valium, tilt the balance of excitatory and inhibitory influences in the brain toward inhibition, thereby causing the incoordination and sedation. Because alcohol produces similar effects, it also likely promotes neuronal inhibition through GABAA receptors. When using depressants such as barbiturates and benzodiazepines, the central nervous system depression occurs due to inhibited secretion of certain excitatory and inhibitory neurotransmitters. Effects range from mild calming to sedation to loss of sensory sensation to cardiovascular collapse leading to death. Benzodiazepines increase either the activity or release of GABA, as benzodiazepines bind to the GABA receptor. They do this on a location separate to where the GABA itself binds and exert an influence over GABA binding. This is known as an allosteric effect, which causes an increase of action at the GABA receptor. Several studies have suggested that changes in GABA receptor function contribute to the tolerance to and dependence on alcohol. Therefore, more alcohol needs to be consumed to produce the same effect, which can lead to the development of alcohol dependence. Combining both alcohol or barbiturate drugs with a benzodiazepine, such as Valium, would create a synergistic effect, significantly increasing the risk of overdose, especially in someone who had built up a tolerance to either drug. Evidence from Malcolm also suggests that long-term alcohol use and withdrawals permanently alter GABA function and downregulate benzodiazepine binding sites. Therefore, chronic alcohol administration induced tolerance not only to alcohol, but also to other substances affecting the GABAA receptor. The chloride channel 1 is a negative chloride ion channel responsible for around 80% of the resting membrane conductance. In inactive muscle fibers, this channel is mainly found in the sarcolemma membrane of the skeletal muscle. The chloride channel 1 plays an important role in membrane repolarization following muscular contraction. Chloride channel 1 forms an ion channel that controls the flow of negative chloride ions into these cells. The main function of chloride channel 1 is to stabilize the cell's electrical charge, enabling muscles to contract normally. Channelopathies are a group of disorders resulting from the dysfunction of ion channels located in the membranes of a cell. When chloride channels become blocked, there is a reduction in the amount of chloride ions flowing into a muscle cell. Myotonia is the most common skeletal muscle channelopathy and is caused by mutations in the skeletal muscle voltage-gated chloride channel gene. Due to a mutation in the chloride channel 1, people with myotonia do not get enough chloride ions admitted into the cell. This shortage of chloride ions causes prolonged muscle contractions, which are the hallmark of myotonia. That is all for my presentation today. I have left a copy of the references in the video description. Thank you.